So we've got today co-occurrence, and next Tuesday is a reading day, so we're not going to have class. And then next Thursday, a week from today, we're going to talk about pie crust. Hopefully I can get it working. Pie crust was working on Snyder, but I've got to get it working on Bell. And then your written projects are due coming up on the 19th. I think I have the same due date on both. On the 19th. So you've got 2 more weekends, I think, is what that works out to be. Now, I've been fairly flexible on due dates. I think probably most everyone has turned in something late. I just knock off a couple of points for late work. But this written report has to be turned in on time. Because after you turn it in, I have to redistribute for peer review. So there's no wiggle room. You have to turn it on the 19th. And then, so your job is then to read someone else's paper and provide comments. And then that'll be due the next Monday. There will be nothing else due in the meantime. Really, there's almost no more assignments. We're not going to have any real assignments on Mother. Even if we're not even really going to talk about Mother. We're going to switch this to the other Chime tutorials. So what does that mean in reality? Like, you have to turn in a paper on Monday the 19th. It may not be the thing that you're the most proud of. There would be time to continue working on your paper, right, in the meantime, and reading one of your fellow classmates' papers. And then the final thing is due May 1st. So the grading will be lighter on the first manuscript. So really you've got a month until the end, so it's the last push to the end. And then actually the final manuscript I have written here is May 3rd, so that's coming up. A few things that you may have forgotten about the final paper. Where is it? Boop, 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 boop. It's so little. How to make this bigger? That's better. Okay. So you do need to have a background section for your paper. It doesn't have to be 5 pages long. You know, did I put a page? 1 or 2 pages, is all you need. You don't need a really, really in-depth background. But we definitely you need to write, why is this important? Why does it need to be done? What's been done before? What still needs to be known in this area? You do need to keep bookkeeping on the sequence filtering. This is your stats. There's an output of data too called something stats.qzv. That's basically fulfills this requirement. You need to keep track of all of your commands. Where do I put this? Where do I have that written? Do I have a rubric? I thought I, sorry, no. Yeah, before all this. You should keep track of all your commands. So hopefully you're building onto some kind of submission pipeline that you've run them. Probably some of them you've run in an interactive session, but then you're saving them into one complete list of all the commands that you've run, especially your chime commands. And so it'd be ideal to have one script where if you had the original data, you could just run for at least all the chime commands. You could run this one file and produce all the output that you then use for our scripts and creating figures later. So that'll be an important element. And I'd like for these eventually to be deposited into a GitHub that you log on, create yourself an account on GitHub, and you basically just need to upload these files there so they can be saved and could be shared, and I can look at it. So that's another part of like the methods part. In your methods, you should show your subsampling depth and why you chose it. You need to have at least 2 metrics of alpha diversity. Generally, we show at least 3. Something to show richness, evenness, and phylogenetic diversity. If some of those are just like totally not interesting, maybe that's one you could leave out, but you need to show at least 2. And then and then also the statistical testing to show if they're statistically different. Beta diversity is the same kind of idea, at least 2 beta diversity measures with statistical testing. The OTU differential abundance testing, and then 2 other methods. 2 out of any of these 4. Random forest, DE-seq, left-C, cooccurrence, or pie-crust. I guess that's 5 choices. And then a discussion section and a conclusion section. Okay. Any questions on that have arisen while you're thinking about writing your paper? Yeah, mine.
What are methods? I mean, I know most of us are getting more studies from already published authors. Do you need to write how they like extracted DNA or?
No. You could give the highlights. Just summarize their methods. We want to know, what primers were used, what's the region of the gene. Like those are important for you and your analysis. Definitely in all the steps that you did, we want to have you can give the details about that. But probably like one short paragraph to give the experimental design, the subjects that were sampled, what kind of samples were taken, and then you should list the statistical analysis that you performed. But no, you don't need to. And there may be other parts of the paper that maybe you didn't use or utilize, so you don't need to talk about things that you're not analyzing. So I would recommend just summarize the most important parts, and then you could say for more details, see the full text and give the reference. Yeah. Okay, good question. Any other questions? Righty. Now today, another thing, I think some have overlooked also the reviews of the articles that are listed on this schedule. So one of the requirements was to write a summary about 5 of these articles that are listed in our syllabus. So make sure you do that. There's not very many people that have done all 5, so make sure to give yourself a chance to finish those off. There's still some papers on pie crust, and the McMurdy paper is a good paper. If you haven't done them yet, feel free, you can go up the list and summarize papers that we've already talked about in class, just to take care of that requirement. Okay, I think that's all of our kind of business. So today we're going to talk about cooccurrence. And I want to bring up the paper. Okay, so I'm not going to go over all the details of this paper, but I'm going to try to hit the highlights. So this is one of the papers on our syllabus, and I think this is quite a good example microbiome study that we should all become familiar with to kind of model our work after. I think it's a great example. It's rewarded. I mean, it's published in Nature, so it's a very good article. And I think part of the thing that makes it very good is that it takes what people normally do, and that's figure 1. And then the rest of the paper is what people usually don't do, and that's what kind of sets it apart. So figure 1 is what we normally do. They set up some different conditions. So this is in mice, and they fed the mice 3 different antibiotics: clindamycin, ampicillin, or enrofloxacin. Do they have? And they fed the mice these antibiotics. I don't remember exactly. For a while, I think it was 10 days or something, just to try to. The goal of the antibiotic feeding was to just establish a different microbiome content. They wanted to establish groups of mice with different microbial compositions. And then they challenged these mice with Clostridium difficile, which is important for causing diarrhea in humans. It's a very problematic pathogen. And then they wanted to know, how well does Clostridium difficile colonize in these different microbiome backgrounds? So that might be what the first part, the first half of Figure 1, may be what most of us usually do. We have some treatment, we apply the treatment, we see what the impact is on the microbiome, and then we look at the results and we kind of think, oh, look, like maybe it changed because of this, and maybe this group is important, and maybe that. So then they applied, on top of that, they applied like a hypothesis, a second treatment. They said, okay, we have these different communities. If we challenge them with a pathogen, in which community will they colonize? And so then that's what's shown here over time. So C. diff colonized this community fairly well, and then C. diff eventually decreased to fairly low abundance. And this group it colonized also similarly pretty well, but then dropped off and was, C. diff did not colonize at all after day 10. And this community, C. diff never colonized at all. So they couldn't use this data because they didn't have any kind of gradient in which to examine. And I think they focused on the associations in the ciprofloxacin group. And so that's pretty, first of all, that's a first off interesting thing. They have different communities. The antibiotic is no longer being used, but in these different community backgrounds, C. diff is sometimes able to colonize and sometimes not able to colonize. So the second thing then they did is they furthered the analysis, and they looked for bacterial populations that correlated with the abundance of C. difficile. And that's our co-occurrence analysis. So that's our topic today. They did this in a variety of different situations. They looked at outside human data, but I'm going to focus on this side over here. Whoa. There's very sensitive. So they perform like Spearman correlations, and these are correlations with the Clostridium difficile. So these were negatively correlated with C. diff, and these were positively correlated with C. diff. So these negative correlations, if these are present, then C. diff is absent, and if C. diff is present, then these organisms are not present. So that may indicate that these organisms are inhibiting or in somehow not allowing C. diff to colonize, which would be beneficial, right? If we could then feed people these organisms that are going to prevent C. diff, then that may be like a preventative medicine. And then they tested this a few times, and again they found C. scindens and other Clostridium species that were negatively correlated with C. difficile. And then this served as another hypothesis then to test specifically on these bacteria to use them as a probiotic to feed. I think this is still mice. To feed mice these bacteria, and then challenge with C. diff again to see what happens to the colonization of C. difficile. Okay, stay here. And so again, they found that these organisms did result in decreased colonization by C. difficile. So, and the control, C. difficile colonized high, 10 to the 8th. With their one main candidate, C. scindens, it was pretty kind of noisy and variable. But if they added like the four most negatively associated bacteria, they got even a better response. And they did this under a variety of situations in each of these. And then later they found that these bacteria were responsible in some metabolic pathways, especially associated with bile acids, and then could even further then try to identify what are these organisms doing in the gut that's prohibiting C. diff from colonizing. So I think for me that's a great use of a microbiome survey to then give you a hypothesis. Well, it's a survey plus an assay, I'm going to call it. The survey is surveying the community, and the assay is challenging with another organism. And then to use that to serve as your next hypothesis, to then start another experiment to test your idea even further. So I think that's a great pattern to follow in our studies, and part of that involved this co-occurrence analysis. Now, probably if you do a co-occurrence, it's probably not always going to work out so beautiful like it did for this group, but I think it's a great place to start. So let's talk about how we can do this. I'm going to plug in my computer. So they used a fairly simple Spearman correlation coefficient. So anybody can do a correlation, right? Now Spearman there's different kinds of correlations, right? A correlation is going to give you something. You're going to look at your data and see if there's a trend here that they follow. And this may be C. diff and ASV 10 or something. Now, does anybody know the difference? I can think of, so they use Spearman, Pearson is another one. These are the two main correlation methods that I think about. Does anybody know what these are and what's the difference between Spearman and Pearson? You said yes, I know them, but no, I don't know the difference.
I only know Pearson.
Okay, what's Pearson?
I just remember R equals.
Okay, and what? Okay, maybe the details are fuzzy. Anybody? Any stats gurus here? Toby, do you know what's Pearson and Spearman?
I think human kind of runs the defense or something like that, while the Pearson value is the p value.
Okay, good. So yeah, Spearman, it does. It correlates the ranked values. So that's like I was talking about with the Kruskal-Wallis. It doesn't use the actual values. It takes the values and then ranks them, and then performs the statistics on those ranked values, not the actual values. Okay, and then Pearson is the opposite. It uses the actual values. So Pearson would be more quantitative, and this is kind of more qualitative. But then there's also some things to worry about. That same old problem with our differential abundance is also somewhat of a problem with correlations, in that our data is compositional data. So that does result in some problems, and there are just like the differential abundance, there's multiple methods out there for doing these kind of correlation analyses. I'm going to show you one, but there are others out there. I'm going to show you a pretty simple, maybe not the most ideal, but it's simple and fairly easy to use. Now there's another, let me see if I can, I don't think I have an example. Let me, well, let me show you what, do I have Eunice's paper? So then these co-occurrence networks can become fairly complicated. Where is it? We'll just use this example. So this example is fairly straightforward. Each of these dots is representing one, in this case, OTU. And they're only showing how many? They're only showing maybe 20 here, because they're centering their analysis around C difficile. So they're really focusing on anything that's positively or negatively correlated with C difficile. But you could imagine if in your data set you had, I don't know, 1,000 ASVs, and then you're correlating them all with each other. So you're doing the entire community network. There's a potential for 1,000,000, 1,000 times 1,000, 1,000,000 correlations, which then becomes really complicated, and it's too much data to understand what's going on. So there's also another method. I've forgotten what the name of it is right now. That it does a so instead of showing these interactions one on one, there will be groups of organisms that are all correlated with each other. Let me show you another example. So I think I maybe already showed you this. So this is some work that I did, um, and this is still a pretty simple network. This is only like 50, 50 nodes, not like 1,000. So we can kind of take this in. There's a group of, in this case, these are genes that are correlated with each other. And what you'll notice is there can be clusters of genes that are genes or organisms or ASVs that are all kind of pretty much all correlated to each other. So within this, most of them are, each of the lines indicates if they're correlated with each other. They're all mostly correlated to every other thing inside of this group. And similarly over here, there's this cluster, and they're all pretty much correlated with each other too. So there's another, I'll have to get the name of it to you. So instead of drawing a network like this, where it's individual genes, they may draw it so that there's a network of clusters of organisms or clusters of genes. So there may be these kind of networks, and these may be correlated, but instead of these nodes being one organism, this may be a cluster of organisms. And then you can say, okay, all these organisms are correlated with like either this group or this group. And it's a way of simplifying the network and being able to visualize what's going on in that community. That's kind of a sidetrack, so maybe I'm getting us distracted, but I think what I'm trying to say is there are multiple methods to do this. What I'm going to show you today is not the only method. Okay. Any questions on this paper before we move on? All right. So I haven't given this to you yet. My week's going crazy, and we're gonna work through this guy together. Okay. So just like normal, there's gonna be the beginning portion of setting up our data, reading in our data, getting it properly formatted, and then this second part. We're not going to do all of this. We're really only going to do these two parts. And then there's a loop that's going to run our correlation analysis. And it's going to print us a table, a list of ASVs that are correlated to each other. And then we'll have to decide what to do with that list. So our script is mostly going to be just these two sections. We're going to read in our data, we're going to run the correlation analysis, and then if we have time, I can show you a way to display it. Okay, so not too many packages we need for this. I'm going to start us over here. Get it a little bit bigger. All right. So I'll make this available after class. So we're going to read in some packages. I'm gonna set our working directory. This is my same working directory I've been using before. We're gonna read in the table.qza. It's not necessary to have the rarefied table. You can use the normal table. So we're using the chime to R function, and we need to transpose this data set. So this T stands for transpose. So you can very easily transpose a matrix just with the T function. And we're going to make it as a data frame. So our data set, our OTU table, is going to have our samples in the rows and our ASVs in the columns, like this. And we also need some metadata. So we're going to read in our metadata also. This is again still our moving pictures example. So now we have a metadata. This should also look very familiar. We're going to merge these two things together. We're going to merge together our metadata and our data set into one. So now we'll have all of our metadata rows and then all of the ASV data after that. And then we have to think about how we're going to draw these networks. We could draw one big network for all the correlations between all the sample types. But in any case where you think you have like a fairly significantly different community, you're going to want to create a network for each environment type. So in this data set we have the tongue and the gut and the hands. We probably don't want to make one try to find if things are correlated in the gut and the hand samples, because those communities are very different. So usually I recommend if you have a treatment group that you're interested in, you should probably create a network for each of those separately. So if you're doing the C-section and the vaginal delivery, if you want to do this, you should probably create a network for each case separately. And that's what this variable here is going to be. So I'm adapting this on the go. So this is going to create a vector of all of the unique values from our body site column. So treatments now is nothing. Body site. Body site. Oh, meta. Okay, so we have gut, left palm, right palm, and tongue. So this treatment vector is just a list of these 4 words here. And now we're going to get into running this loop here. We're going to set up this value called final results. It's going to be just an empty data frame. And then we're going to run this loop for values of 1 to the length of treatments. The length of, remember what treatments is, it's this. So what the length of treatments is 4. There's 4 items in this list. Our length of treatments, this is 4. And I need to change just a couple of things here. This thing here, this is the column name of the treatment that you're interested in. So we're interested in body site. And treatment bracket I, that's basically, let me set I to 1. And we'll walk through this. So this line here is going to subset our data for only those that are body site of treatments 1. Treatments, so what's treatments bracket 1? Anybody remember this from R? What's this asking for? Remember, I is just 1 right now. So what's this going to add? What's the value of treatments bracket 1? Add one of the, it's not going to add one of the treatments, it's going to get the first one in the list, which is gut, the first one. If I was 2, then it would get the second element in the list, which is the left palm. And then it's going to then subset the whole data set so that we only get those that are, the first time around, those that are gut samples, and the second time through the loop, those that are the right palm samples. So this temp thing here is going to get just the gut samples. Okay, so our table now is pretty small, just the gut samples. That's another thing here, or another warning, is in order to create these networks, the more samples is always better. But you should definitely not try this at all. Definitely not at all. If you have less than 6 samples, don't even try. That's too few samples. This example we have 8. That's also very, very few. So for this kind of correlation analysis, really it's better to have more than 20 would be better. So it is sensitive to having too few samples. But we've got 8, so that's all right. So we're going to make this correlation. The next thing here is where a good deal of the work happens. This rcore function, this is what runs the correlation. And this is a special tool where it's expecting that you give it a matrix, a data frame of many, in our case it's many ASVs, and it's going to perform all the pairwise correlations between all of those columns. So they need to be in the columns. So it's going to correlate column 1 and 2, and 1 and 3, and 1 and 4, 1 and 5, and then 2 and 3, 2 and 4, 2 and 5, and then 3 and 4, and 3 and 5. It's going to do all the pairwise correlations. And you can give it different metrics to use. So you can use Spearman. You can also use Pearson. We get help with this.
So I guess it's just we want to use.
Pearson or Spearman. Yeah, I would recommend Spearman. That's going to help with the compositional nature of the data. So if you're just using this ASV data, this 16S sequencing data, I wouldn't recommend Pearson. I would recommend the Spearman. This little thing here, I should add a comment. Use the -c parameter to remove metadata columns. So this -c bit here, we need to remove all of the metadata columns. Kind of like the left C analysis, if you have metadata, it's not expecting to have any metadata columns. And if you do, then it gives you an error. So in our temp item here, we need to remove columns 1, 2, 3, 4, 5. We need to remove all of these up to column 9. So if we run just this, temp, so temp is going to keep, before the comma are the rows, it's going to keep all the rows, and it's going to delete columns 1 through 9. It's a big long thing. It's too much to look at. Let's do it a different way. So this is that one small section of that line. It's deleted all the metadata. So now all we have are our ASVs data and no metadata. So now we're set. We can correlate this column with this column because it's all values, numbers. Okay. So this line here is going to do all that work. It's going to do all the pairwise correlations. I don't know how long that's. That's fast. So the result of this correlation function is this results thing, and this is a complicated element. It has 3 items in it: R, N, and P. So the R item is a matrix of all of the R values, the row, the correlation coefficient. The N matrix are all of the N, the number of values that went into each correlation. And the P matrix is the P values for each correlation. So if we look at this, so the R element of this results thing that we just made is just the row values. So the correlation coefficient between the first ASV and itself, the correlation coefficient is 1. They're the same values. And the correlation coefficient between ASV 1 and 2 is this, and 1 and 3, and 1 and 4. So we have all of those correlation coefficients for all the pairwise correlations. And then we can get all the p-values. And we're going to go through a few things here. We don't need to go through all the details. This is going to reformat the p-values into a long list. So instead of a matrix, you've got ASV1, ASV2, and then the p-value listed. This one is going to calculate a q value for those p-values. I want to change this. So now it's adjusting our p-values to q-values because we have so many p-values, right? We have, this thing is 34,000 items long. So there's definitely a multiple test problem here, so we need to adjust those p-values. It increases the values pretty significantly. So that's a good thing to prevent false positives. I'm just going to change some names in these data frames. And then this step is going to select anything with a Q value less than 0.05. So we're going to start filtering our data set. Rows melt. Then we're going to convert our row matrix to a data frame also. We're going to merge the P's and the rows together. So now we're going to have a merged item here that has ASV1, ASV2, the P value, the Q value, and the row value. And then merge, and we're going to list the treatment in a treatment TRT column. The treatment is the gut. So we know that these ASVs are correlated in the gut. And then we're going to save this into our item called final results. And we're going to print a message called we finished the gut. Okay, and so then we'll end up with a table called final results that has that result table we're just working on. ASV1, ASV2, the P values, the Q values, the correlation coefficient row, and which sample they were found in. Now, it would also be useful to have our taxonomy to add to this. Because that is this very simple method of doing this correlation analysis. So we've just run it for the gut samples. Then this loop, if we ran the loop all together, then it would run it for the left hand, the right hand, and the tongue samples all in sequence. So we can run that. So it's running for the gut, the left palm, the right palm, and then the tongue. And then in the end we have a final results. That's just like before, except now it has the gut samples are the top, and then the right palm is listed. The left palm is in there somewhere. Left palm. And then the bottom are all the results from the tongue. It just seems like there's not as many correlations in the tongue. There's some here. Okay, so any questions on what went into making that results of the correlation analysis? All right. Okay. Now, there are some other things to worry about here. The correlation, a lot of times these correlation programs are they're not as well developed. So something to be worried about is if this is ASV1 and this is ASV2, it could be that these two ASVs are pretty rare. Maybe they're absent. Their abundance is 0. And like samples 1 through 7, they're all absent. And in sample 8, they both happen to be present. They could have any abundance. It doesn't matter what they are. It's basically a straight line between these samples where it's absent and if there's only one sample where they both happen to be present, then they're going to be it's going to be a perfect correlation, right? So and it doesn't matter. It could be completely meaningless. It could be anything out here could be a perfect correlation if they're mostly all absent and most of the samples, and just one sample they happen to be present. Now, if they're mostly absent and then they're present in 2 samples, this is going to be a lower correlation, and the p-value may not be significant. But you could imagine that if they're absent in no samples and they line up something like this is going to be a correlation that we're much more interested in compared to something where they're absent and only present in one sample. This is not really meaningful, and this is meaningful because they're actually there, and over all 8 samples, there is a correlation there. So we can control for that in this script with that in. Where? I have to find my other example, but I don't think I'll be able to throw that in here on the fly. But I'll have another example. It's basically almost the same thing, except we add in, just like we use the row values and the p values to filter to make this final results item. We can also use the n to tell it how many samples were these 2 ASVs present. So usually I like to introduce a filter where the n needs to be greater than like half of the samples that are in the experiment, or at least in this case, we only have 8 samples. So it'd really be best if n was greater than 6. Because if it's just 2 samples too, it could just by chance that they lined up rather than actually being correlated. So that's another thing that I would warn about, because you don't want to get a bunch of these spurious correlations that are really just random chance. Okay. Any other questions about that? All right. And then there is a software to visualize networks called Cytoscape. Has anyone ever used Cytoscape before? It's a free program. It's a point-and-click program. So this network that I made, I used Cytoscape program. It's pretty useful. So it's a free download. Let's see if I can make this bigger. It's all very small. Okay. Import. So we need to give this, we need to import some data. And I've got, so this is an example from before. Strong results. I've got lots of different iterations of, let's use this one. So strong results. So this comes from the function I just used. Our end thing was called final results. Let me delete some of these things here. So oftentimes it's there can be a lot of results. So the results we're getting from this has 42,000 correlations. It's just a lot of data. A lot of them could be weaker correlations. So like if the rho value is too low, actually most of these are, I think our rho values are high because I didn't filter out based on the n. But you may think, well, I've got 42,000. I'm only interested in the most strong correlations. So you could then filter this and say, okay, I'm going to require a rho value of 0.9 or greater to make the data more amenable to be visualized and digested. Or you may choose a smaller Q value. So you may say, okay, the Q value of 0.05 is not stringent enough. You could rerun the code here and change the Q value to be maybe 0.1. Let's see.
Rho, they're just showing how fun correlation is.
It's the correlation coefficient, yeah, both negative and positive.
Could you make it? Could you filter by you just want negative correlation?
You can filter if you just want negative correlation, positive correlations, right? Like so, This is going to have a very small or very high row. This might be a row 0.98 or something. And if there's more noise here, this might be a row 0.6, right? So it's the how variable the data is around this line of best fit. So you can play with how you want to filter the data set. So like the Clostridium difficile paper, maybe you're only interested in negative correlations, and maybe you're only interested in correlations with an ASV that is C difficile. So then you could also filter it that way. Okay. This is better. It's really small, right? Import a network from a table. So you need to indicate what are nodes. So some more vocabulary. And in these networks, we have nodes and edges. The nodes are the dots, and the lines are the edges. These are nodes. And this is an edge. Okay. And we need to tell Cytoscape what is a node and what is an edge. So our ASVs, those are the nodes. So this says source node. You may not be able to read that. Source node. So we don't have a source and a target. We don't have any directionality. They're without direction. They're just nodes. So, but we'll label one the source, and the second ASV is the target. The p-value relates to the correlation, so that's going to be a edge attribute. So it's an attribute of the edge because it describes the correlation between the two. So the p-value, most everything else is going to be an edge attribute. P-value, Q-value, row, those are all edge. Treatment. Treatment is all the same. This would really be more of a, well, we'll call it an edge attribute too. Could not finish the layout. Okay, I'm trying too much on the fly. Something is going wrong. We can quickly finish this up the next time. Okay. This is just, but the Cytoscape is just to visualize the network, so you can definitely do the first steps without the Cytoscape part. So we'll have to end there today. I will pick up the rest of Cytoscape the next time, hopefully. And you don't necessarily have to draw the network. You can just look through your table for the things that you're interested in, to find correlations between one particular ASV and other things that may be correlated with it. So, all right, guys, that's where we'll end today. So let me know if you have questions about anything. But then won't see you till next Thursday. So enjoy your reading day. I'll see you next time.
Is there something different tomorrow for the project?
Tomorrow?
Yeah.
Yeah, tomorrow are any two, so it's two of the extra analyses.
Yeah, I don't see a submission.
Oh, the place to submit. Yeah, I need to create a submission portal. Yeah. So that would be any of these: DE-seq, left-C, random forest, or cooccurrence. Any two of those on your project data.
I was there last week. Oh, was it last week?
What was it?
Yeah, that was last week.
So DE-seq, well, just DE-seq. So I guess you only need to really do. I guess I kind of changed things. So you would just need to do one extra one, I guess. Choose left C or random forest or cooccurrence to perform, and that would be due tomorrow. Because yeah, DE-seq was due last time. That sound all right? So just choose one of those other three.